Welcome again. Today we continue to work with this table of messenger RNA codons, looking at how to work with it now in the reverse. So here we have the table of messenger RNA codons, but the objective in this question is to determine the original DNA sequence, also known as the antisense sequence, that produced this sequence of amino acids and start and stop codons. So we have a start codon here, we have a stop codon here, and we have these amino acids with these short names here of the amino acids: leucine, glutamine, tyrosine, proline, and valine. All of these amino acids included between the start and the stop codons. So the first thing you would need to do is to go find the start codon, and then going in reverse, we see that you have an A, a U, and a G. For the first letter, it's an A, second letter, a U that comes down here, and the third letter that comes into this box is a G. So then AUG is what gives the start codon message. But what does that correspond to in DNA's sequence? Remember that A, adenine, pairs with T, thymine, and U, uracil, pairs with A, adenine, and G, guanine, pairs with C, cytosine. So in this way, you would determine the corresponding DNA sequence, the one that's on the antisense strand, the one that the messenger RNA actually binds into to line up and form its sequence. The other strand is referred to as the sense strand, and that one would have the corresponding DNA bases that match the RNA sequence. And when I say the corresponding DNA bases, I say that because you need to remember that in DNA you do not find uracil. Thymine is replaced by uracil. So I would like you now to go ahead and to finish the rest of this sequence. Remember, you're focusing on the DNA strand that the messenger RNA would lock into or fit into. That is called the antisense strand. The other DNA strand is called the sense strand. So when you're finished, you could see your answer to this question in the caption below this video.